Hello, this tutorial is about the biomolecules, or what is called also the chemical building blocks of life. In order to start, all the biomolecules are made out of carbon, so carbon is the central element. The biomolecules contain carbon in both chains or rings. Remember from the first class of chemical bonds that the carbon has 4 outer shell electrons or 4 valence electrons. It means that with these 4 that we can see here in the Bohr's model, you can make the chemical bondings. So this enables the carbon to make chemical bonds with itself and with other elements. Therefore, the capacity of bonding of the carbon is great, and that's why it's the central component of all the biomolecules. All the bondings that the carbon is making are covalent, so hence it has strong bonds. Well, all the biomolecules are organic compounds, meaning that they are all made of carbon and that may include hydrogen and other elements. Inside the organic compounds, you must find the carbohydrates, lipids, proteins, and nucleic acids. And also you may find some vitamins. The types of biological molecules that we are going to talk about are proteins, lipids, nucleic acids, and finally the carbohydrates. To know what a biomolecule is, you have to take into account that all biomolecules are also called macromolecules. Macro, as its name states, is a large biomolecule. Many of these biomolecules are polymers. But what is a polymer? So a polymer is a long molecule built by linking together small similar subunits. These subunits are called the monomers. The macromolecules are the sum of the parts. So the macromolecules, as they are polymers that are the result of linking together monomers, have the function only if they are joined together. Otherwise, you are not going to have the chemical function of each one of the biomolecules. Let's see the main monomers that made up the different types of biomolecules. For example, proteins are polymers made up of amino acids. Nucleic acids are polymers of nucleotides. Starches are polymers of simple sugars that are called the monosaccharides. How can I construct or build up the polymers? In order to go from just monomers to the composition of polymers, you need to take out this kind of chemical reaction that is called the condensation polymerization, or also dehydration synthesis. In this kind of condensation process, what you are going to do is to form a water molecule. Okay, so you are going to extract water in order to form the chemical bond between the two monomers. In order to do so, you need to use energy. Remember that this cell uses the energy in the form of ATP, that is a nucleotide that is carrying energy. So the total reaction is like this. I have this monomer in here, I have the other in this side, and I am going to use this hydrogen ion plus this hydroxide ion in order to form the water. I'm going to extract this molecule out using the ATP, and then I have the formation of the new chemical bond. So in this way, I have made out the condensation polymerization, and I just formed the new polymer. This is the diagram that I just explained. You start with monomers, you use the ATP. The ATP is going to give you the energy to split the water out and to form the polymer at the end. Also, in nature, you are going to require to break down the polymers that are already made. So the opposite to the condensation reaction is called the hydrolysis. In hydrolysis, you break a covalent bond by adding the water molecule. In this case, you are not going to use ATP. This is going to occur in a spontaneous way. Then you have the polymer in here. You add the water molecule, and this water molecule is going to break down this covalent bond by adding one hydrogen ion in one side and the hydroxide ion in the other. The general diagram is this one. Is you start with the polymer, you have the input of water in order to break down the covalent bond that already exists, and finally you have the monomers by the side. Now let's take a look to the different types of biomolecules. We have the carbohydrates, the lipids, the proteins, and finally the nucleic acids. Then start with the carbohydrates. Carbohydrates are, for example, these ones that are the starches that you can find them in this case in the chloroplast, but also you have others like the glycogen in animals or cellulose also in plants. In this slide, I am having the cellular structure that contains each one of my biomolecules. In this case, the chromosome that is containing a DNA strand that is a nucleic acid, and in here I have the monomers. In the case of the nucleic acid, is called a nucleotide. The cellular structure that contains proteins, in this case, the example is a muscle fiber, the polypeptide, that is the polymer or the protein, and the monomer, the amino acid. Finally, the cellular structure that contains lipids, in this case, the adipose tissues. My polymer is called the triglyceride, and my monomer is going to be the fatty acid. What's the basic function of each one of these biomolecules? The carbohydrates are used to store energy, but this energy is of short-term use. It's an energy that is going to be used as soon as you acquire it. And also some of them are structural. Some examples of the carbohydrates are the starches, glycogen, glucose, sucrose, and cellulose. Let's move to the other group that are called lipids or the fats. The function of the lipids is for long-term storage of the energy. In contrast with the carbohydrates, this storage is of long-term rather than an immediate use, but both are storing energy. Also, lipids have an insulation function and a protection one. The examples inside lipids are the fats, oils, and waxes. The next group are the nucleic acids. They are the ones in charge of the inheritance information, and they have the blueprint for metabolism. It's compounded by the DNA and the RNA. The DNA for deoxyribonucleic acid and the RNA by ribonucleic acid. In this square, I have the ATP inside the nucleic acid just because it's a nucleotide, but it doesn't mean that it's carrying out the inheritance information. Remember that the ATP is a molecule that is going to be used inside the cell for giving up energy in order to make the chemical reactions. The last group are the proteins. Those are serving as catalysts, as hormones, or in a structure. They made up the proteins and also the enzymes. The enzymes are the proteins that has this function of catalyst. We are going to see later what is the function of a catalyst. Let's start with the carbohydrates. Carbohydrate means hydrated carbon. These molecules are made up of carbon, hydrogen, and oxygen mainly. They have a specific ratio of 2 to 1 in hydrogen and oxygen. They could be simple sugars or simple monomers like glucose, or otherwise they can be complex polymers like cellulose or lignin. Inside carbohydrates, I have the regular sugars that could be monosaccharides or disaccharides. It means made up of two different monomers. In the other branch, I have a huge group that are called the polysaccharides. Those are the huge polymers like the starches or the complex sugars. The monosaccharides are sugars made out of only one ring. Okay, they are not joining together to anything, they are just existing as units, like the fructose, the galactose, and the glucose. Okay, here I have the normal glucose formula. You see that is a ring, it's not attached to any other thing. The difference with galactose, as you may see here, is the position of some chemical groups in these bonds. What is the importance of the monosaccharides? That they are the energy source that is used as a reactant in the cellular respiration. Also, if you have monomers, you can start the formation of dimers and the polymers by the condensation reaction that we already saw. The next group are the disaccharides. D meaning two, so in the case of lactose, it's the joining together of one unit or one monomer of glucose plus galactose, the maltose, that is glucose plus glucose, sucrose, that is glucose plus fructose. In the case of the formation of the disaccharides, we are going to make a chemical bond. That is the one that I am showing here. We are going to see that each one of the biomolecules have a specific type of chemical bond that gives a specific characteristic to the biomolecule. In the case of the carbohydrates, the name of the chemical bonding that is specific to this group is called the glycosidic linkage. The glycosidic linkage is going to be found then in any kind of sugar. This one that is being depicted here is an example of a glycosidic linkage between one unit of glucose and one of fructose. In this case, we are taking a look to a molecule of sucrose. This is an example of how the disaccharides are formed by the condensation reaction. So I have my unit of glucose that is going to be joined together to this other monomer of glucose. The part that is in blue is the part that is going to make out the water molecule. Remember that in condensation, I need to extract water in order to make the chemical bond. So this part in blue is the water that you can see here at the end of the reaction. And here in red, you are looking at the chemical bond, the glycosidic bond that was formed in the case of maltose. And also, in order to form the lactose, that is the sugar that you find in milk, you need to use 1 unit of galactose plus 1 of glucose. Once again, through condensation, you take out the water, you put it in here, and you have the formation of the glycosidic bond or linkage. Where can I found this kind of sugars? Inside, for example, sugar cane, inside sugar beets, and they are basically storing the energy. And this energy is stored in the chemical bonds. Let's move to the polysaccharides. They are huge sugars. They are quite complex. They could be, some of them could be linear, but most of them have little branches and turns over that makes chemical characteristics. They are very resistant. They are difficult to digest because of the many glycosidic bonds they have. Some of them are having a function in the structures, like for example the chitin. Chitin is a structural carbohydrate that makes out the exoskeleton of many animals. Okay, so in this case we are looking at other function rather than storing energy. They are giving support to this animal. Let's move to the next biomolecule. They are called the proteins. Proteins are made out of carbon, hydrogen, oxygen, nitrogen, and sulfur. They are made out in the ribosomes. The ribosomes are small factories in the cell that takes the information that was coded in the DNA, and through a process that is called transcription, they help the cell to understand the message from the DNA. How? Making a molecule that is called a messenger RNA. This messenger RNA is going to be translated into a language that is the language of the protein, that are the amino acids. Here we're looking at the ribosome. You can observe, you can watch the messenger RNA here. And after the translation takes place, you have the formation of the protein that actually is a chain of amino acids linking together. The name of the specific chemical bond inside the proteins is the peptide bond. Remember, in the carbohydrates we were talking about the glycosidic linkage. So in the proteins we are going to talk about peptide bonds or peptide linkages. The proteins are made out of chains of amino acids joined together. So the amino acid is the monomer in the protein group. This is the general structure of the amino acids. You have an amino group, that is this one. You have a carbon that is central, and a carboxyl group that is at the end of the molecule. This structure is going to keep constant in the 20 naturally occurring amino acids that we have. However, take a look to this R. The R is for radical group. This R group is going to be very variable in each one of the 20 and is the responsible of the identity, or the chemical identity actually, of the amino acid. Depending on this radical group, you may find hydrophobic amino acids, hydrophilic. You are going to have positive charge, negatively charged amino acids. And these chemical characteristics are going to define the final structure of the protein. So all of the amino acids have the same basic structure except for the radical group, that is the one that is going to define the chemical identity or the chemical properties. Here are the 20 amino acids that we may find in nature. As you see, here are the radical groups that are quite different in structure, and that's why the proteins fold and have so different functions depending on the interactions. Let's take a look to the folding of the polypeptide to form the protein. When you talk about the proteins, it's quite important to take into account the shape. The shape is vital for the protein because it determines how they interact with other molecules, and it also determines the particular function of the protein. If the protein is not folded in a proper way, the protein is not going to work. In order to be folded, the protein must follow different steps. We start with the primary structure, that is the one that is given by the ribosome just by interpreting the DNA code. So as soon as the ribosome is being producing the protein, you have a chain of amino acids linked together. It's like a line. This is called the primary structure. In this structure, you don't have still the function of the protein. As soon as you have the primary structure, these amino acids are going to start like interacting with each other because of the radicals they have. So you're going to have some amino acids that are going to be attracted toward other ones, and others that are going to be repelled because of the forces they have. These lead to the formation of the secondary structure. In the secondary structure, you are going to find alpha helixes and beta sheets that are forms of folding in the secondary structure. The secondary structure is still not having the function of the protein. The function is acquired in here, in the tertiary structure. The tertiary structure is defined by the disulfide bridges. Some amino acids have sulfur. The sulfur is attracted to another sulfur. So no matter how long they are, how far they are in the molecule, the sulfurs are attracted so strong to each other that they form disulfide bridges. This is going to give a three-dimensional conformation in the protein protein, and the protein is going to lose the function. Remember that the shape is very important in the function of the protein. If you lose the function, if you lose the shape, you lose the function immediately. So if you break down the tertiary structure just by breaking down the disulfide bridges, you are losing the function of the protein. Most of the proteins just stop the folding here in the tertiary structure because they acquire the function. However, there are some other proteins that go further to the quaternary structure. In the quaternary structure,